Hi everyone, I'm Xiao Luo from Bielefeld University. I will present a new approach, phasebook, which is used for haplotype-aware de novo assembly of diploid genomes for long reads. I will talk from the following aspects. First, I will talk about the background of this project, the method we developed, and some benchmarking results, and make a summary. So what is diploid genome? A diploid is an organism which has paired chromosomes, one for each parent. Most eukaryotes are diploid. For example, human genome is diploid genome. One copy is from father and the other is from mother. Determining the DNA sequence of each copy is called haplotype-aware genome assembly. Haplotype reconstruction plays an important role in various disciplines. Firstly, haplotype information is important in functional genomics since there is a widespread allele-specific gene expression across the human genome. Secondly, the haplotype-specific combinations of genetic variants usually affect disease phenotypes and clinical responses, which is important in precision medicine. Thirdly, it can also be used to access population demography, gene flow, and selection in conservation genomics. Over the last few years, long-read sequencing technologies improved genome assembly a lot because of their long read length. Generally, the long read length is from several kbs to hundreds of kbs, or even to megabase pairs, such that they can span the complex repeat regions in the genome. There are many too-long read sequencing technologies, PacBio and nanopore sequencing. There are two kinds of reads of PacBio, hi-fi reads and CLR reads. The main difference is that CLR has high sequencing error rate, for example from 5 to 15%, which is similar with nanopore reads, whereas hi-fi reads have less than 1% error rate. So the existing methods for haplotype or genome assembly of diploid genomes for long reads basically fall into two classes. The first one is reference-based methods such as WhatsApp, HapCart2. They make use of a high-quality reference as backbone and align reads to it to call variants, which however introduce reference biases. The other one is de novo assembly-based methods such as vacuum, Kalu, Fly, but most of the current de novo assemblies are usually prone to collapse homologous sequence into one consensus sequence, thus fails to capture the haplotype information, or some of them are only specialized for hi-fi reads, such as hi-fiism. So as far as we know, there is no method that firstly generates high-quality haplotype-aware genome assemblies only based on long-read data, and secondly is capable of handling both PacBio and nanopore reads, but do not depend on high-quality reference as a backbone at the same time. So our work contributes to bridge this gap of these related works. We developed a novel approach, phasebook, for haplotype reconstruction of a diploid genome in a de novo paradigm by only using long reads. From a large perspective, phasebook uses a divide and conquer strategy. In the divide stage, it separates reads into small local groups, which results in many local instances of the minimum error correction problem. Solving the local minimum error correction problem by using, for example, WhatsApp concludes the divide stage. Notably, solving the local minimum error correction problem does not need high-quality reference. It works well already using a local coordinate systems based on the overlapping reads in small read groups. In the ends of this stage, we generate phased and error-corrected reads. And in the conquer stage, it generates assembly output using overlap graph-based strategy, which is much easier. As such, phasebook reflects a combination of an approach that is able to solve the minimum error correction problem without reference sequence and performing overlap graph-based assembly in a Diploid-aware setting. So this is the workflow of phasebook. Briefly, our approach firstly splits raw reads into small classes based on read overlaps and separate reads into two haplotypes in order to obtain haplotype-specific consensus sequence, which are also called superreads. So superreads are error-corrected and longer sequences. Secondly, phasebook constructs an overlap graph where nodes correspond to superreads and edges indicate that the superreads stem from identical haplotypes. Based on the resulting overlap graph, phasebook extends superreads into haplotypes as the final output. Let's get some more details. First of all, reads are sorted by descending order of read length, and we select the longest read as a seed read, and then collect its overlapped reads. As we can see here, the color of the line corresponds to the haplotype. So the overlapped reads could be from both two haplotypes because of sequencing errors. All these reads form a read cluster and marked as used reads. And in the next step, we select the longest read from the unused reads as seed read and collect its overlapped reads, which forms a new read cluster, and then do this iteratively until no read cluster generated. Finally, we will generate many read clusters. So for example, this is a read cluster we introduced in the previous slide. The read fork represents the sequencing errors. For a read cluster, we assemble these raw reads into an ad hoc reference and map these reads back to the reference. Then we can find out the true mutations using probabilistic model. Here we use the long shot. The triangles represent the SNPs. Then we phase the reads into two groups by solving the minimum error correction problem. Each of read group belongs to one haplotype. Here we use what's hap to phase them. So within each read group, we generate haplotype-specific consensus, which is also called superread, because both ends of the superread cannot be corrected because of a low coverage. We could set a threshold to trim both ends of the superreads. Thus, for each read cluster, we could get two error-corrected superreads. So the next step is to assemble the superreads. For example, these are superreads in the top area of this figure. First, we calculate the overlap between the superreads and filter overlaps with a more strict threshold. Then we construct a superread overlap graph, which the node is a superread, and the H means that there is a reliable overlap between the two nodes, such that the two nodes are probably from the same haplotype. Then we simplify this graph by removing transitive edges. There may be still a few spurious edges in the graph, such as the red arrow line here, which represents a spurious superread overlap from different haplotypes. Then we find all simple paths in the graph and then concatenate the sequences in a simple path to generate a haplotype-specific contig, which is called haplotype. Finally, we get all haplotypes as the final output. We evaluate our approach on both simulated and real long-read sequencing data. For simulated data, we use 2 human MHC haplotypes to construct a pseudo-diploid genome. For real data, we use human chromosome 6 of these 2 individuals. We test on all 3 types of long reads. And the assembly performance was evaluated by several commonly used metrics, which were reported by cost. For example, haplotype coverage is the percentage of land bases in the ground truth haplotypes covered by haplotypes, which is used to measure the completeness of the assembly, and both M50 and NGA50 are used to measure the contiguity of the assembly, and the error rate is equal to the sum of mismatch rate and the error rate when mapping the obtained context to the reference haplotype sequences. And the misassembled contig proportion is equal to the number of misassembled contigs divided by the total number of contigs. So this table shows the benchmarking results for PAC-by-wall hi-fi rates. The top region shows the results for simulated data, and the bottom region shows the results for real data. In summary, the hi-fi sets the standards on this hi-fi data, and phasebook is comparable with hi-fi in terms of haplotype coverage, error rate, and misassembled contig proportion. In addition, for PAC-by-wall CLR rates, phasebook largely outperforms other methods in terms of haplotype coverage on both simulated and real data, while keeping better or comparable performance in terms of other metrics such as error rate, misassembled contigs proportion. While for M50 and NGA50, phasebook shows comparable or worse performance than others. And for nanopore rates, phasebook also outperforms other methods in terms of haplotype coverage, while keeping better or comparable error rate. We also evaluated the runtime and memory usage of these related tools on both simulated and real data. Briefly, our approach shows comparable or slightly worse performance compared with others. So this is for the PAC-by-wall hi-fi rates dataset. And this is for PAC-by-wall CLR rates. And this is for the nanopore rates. So in summary, we present a novel approach, phasebook, for haplotype reconstruction in a de novo paradigm of diploid genomes by only using long reads. Benchmarking experiments on both simulated and real data demonstrate that our method drastically outperforms other approaches in terms of haplotype coverage, while maintaining better or comparable performance in terms of various other aspects. Therefore, the integration of phasebook in population-scale long-read sequencing projects may open up novel opportunities. In further research, the key idea of phasebook could be extended to polyploid genomes. This work was carried out with the help of these people, First of all, my supervisor Alexander and another PhD student, Xiong. Both of them are from Benefit University, and Yasmin from Harvard Medical School, and Marlin from Tilburg University, Tobias from University of Düsseldorf. Thank you for these people, and thank you very much for your attention.